Welcome to this lecture on steroid hormone receptor families. Today, we are going to explore one of the most fascinating communication systems in the human body. We will learn how specific hormones act like master keys, bypassing the outer walls of our cells to directly rewrite our genetic instructions. By the end of this session, you will understand not just the biology of these receptors, but also the mathematics that govern their behavior, which is the absolute foundation for modern pharmacology and treating complex diseases. To begin our journey, let us imagine your body's cells are like highly secure houses with strict rules about who can enter. Most chemical messengers, like peptide hormones, are like delivery drivers. They must ring the doorbell on the outside of the cell and wait for someone inside to pass the message along. Steroid hormones, however, are entirely different. They are like VIP family members who have their own keys to the front door. Because steroid hormones are synthesized from cholesterol, which is a type of fat, they can effortlessly slip right through the fatty walls of the cell membrane. Once inside, they wander freely until they find their specific partner. Now that we understand how these hormones enter the cell, let us look at what they do once inside. In endocrinology, the specific partners that steroid hormones bind to are called the steroid hormone receptor family. This family is a major branch of the broader nuclear receptor superfamily. Unlike many other receptors that sit on the outer cell surface, these are specialized proteins located entirely inside the cell. Structurally and functionally, they are known as ligand-activated transcription factors. This means they remain completely inactive, essentially turned off, until a specific molecule, called the ligand or hormone, binds to them. Once activated, they travel directly to the cell's DNA to turn specific genes on or off. Knowing that these receptors interact with DNA, we must examine their physical structure to see how they achieve this. Every steroid receptor is built with distinct functional parts or domains. First, there is a highly variable N-terminal domain, which is responsible for the actual activation of genes. Next is the central DNA binding domain. This region contains specialized structures called zinc fingers, which literally grip the genetic code of the DNA. Finally, there is the C-terminal ligand binding domain. This is the specific pocket where the steroid hormone actually attaches to the receptor. Together, these three domains allow the receptor to receive a chemical signal and translate it into a genetic action. Building on our understanding of their structure, let us categorize these receptors based on where they hang out before the hormone arrives. Based on their resting location and behavior, steroid receptors are generally divided into two main categories. Type 1 receptors wait out in the living room of the cell, the cytoplasm. They float there until a hormone, like the stress hormone cortisol, binds to them, after which they move into the nucleus. On the other hand, type 2 receptors are already waiting inside the master bedroom. They sit directly attached to the DNA in the nucleus, waiting for their specific hormone to arrive and flip the switch. Both types ultimately achieve the same goal of altering gene expression. With the mechanics of these receptors clear, we must ask why they are so vital to human physiology. Understanding steroid hormone receptors is absolutely critical because they control the most fundamental long-term changes in the human body. Unlike quick-acting nerve signals that last for mere milliseconds, steroid receptors alter gene expression. This means they literally rewrite the daily instruction manual of the cell, a slow but powerful process that unfolds over hours or days. Because of this profound level of control, these receptors govern major life events like puberty and pregnancy, as well as your daily metabolism and your body's response to severe physical stress. Because these receptors wield such immense power over cellular function, they are incredibly important in modern medicine. In the real world, these receptors are the primary targets for some of our most important and life-saving drugs. For example, certain breast cancer cells hijack estrogen receptors to grow uncontrollably. To combat this, doctors prescribe medications like tamoxifen, which specifically bind to and block these receptors, effectively starving the cancer. Without a deep understanding of these receptor families, we would have no way to treat severe inflammatory diseases, hormone deficiencies, or hormone-dependent cancers. This medical reality makes mastering their behavior essential. To truly understand how these life-saving drugs and natural hormones work, we must look at the mathematics of binding affinity. The biological effect of a steroid hormone depends directly on how many of its receptors are currently occupied. We calculate this using the receptor occupancy equation, derived from chemical equilibrium. In this formula, Y represents the fractional occupancy, which is the percentage of bound receptors, expressed as a decimal from 0 to 1. The symbol L in brackets is the concentration of the free ligand, or hormone, usually measured in nanomolar units. Finally, Kd is the equilibrium dissociation constant. This is a unique number telling us how tightly the receptor holds the hormone. Specifically, Kd is the exact hormone concentration needed to fill exactly half of the available receptors. As the ligand concentration increases, the fractional occupancy Y also increases, leading to a stronger biological effect. Now that we have our formula, let us see this with a concrete example involving the stress hormone cortisol. Imagine a patient experiencing a highly stressful event. The concentration of free cortisol inside a target cell is measured to be 4 nanomolar. The glucocorticoid receptor has a known dissociation constant of 4 nanomolar. We want to find the fractional occupancy. We start by substituting our known values into the equation. We place 4 in the numerator for the ligand concentration, and in the denominator, we add the dissociation constant 4 to the ligand concentration 4. Simplifying the denominator gives us 8. Dividing 4 by 8 yields a final decimal value of 0.5. This means exactly 50% of the receptors are currently bound to cortisol. This makes perfect intuitive sense. Whenever the hormone concentration exactly matches the dissociation constant, the occupancy is always exactly half. Having successfully calculated occupancy, let us tackle a more complex scenario where we work backward to find a required drug dosage. A researcher is testing a new synthetic estrogen drug with a dissociation constant of 2 nanomolar. They want to achieve a high target occupancy of 90% or 0.9 to trigger a strong genetic response. What concentration of the drug is needed? We substitute 0.9 for Y and 2 for Kd. To solve for the unknown ligand concentration, we multiply both sides by the denominator, bringing 2 plus L up to the left side. Distributing the 0.9 gives us 1.8 plus 0.9 times L equals L. Subtracting 0.9 L from both sides leaves 1.8 equals 0.1 times L. Finally, dividing 1.8 by 0.1 gives us 18. The required concentration is 18 nanomolar. Notice that to go from 50% to 90% occupancy, we needed a concentration 9 times higher than the dissociation constant, demonstrating a crucial non-linear relationship in pharmacology. Now that we have explored the mathematics, let us visualize this entire biological process in action. In this diagram, you are looking at the complete journey of a steroid hormone. On the far left, the orange circle represents the lipid-soluble steroid hormone effortlessly crossing the cell membrane. Once inside the cytoplasm, it finds its specific blue steroid receptor. Notice the green molecule labeled HSP90. This is a chaperone protein that keeps the receptor inactive. When the hormone binds, HSP90 detaches. The activated receptors then pair up and travel through the nuclear pore into the nucleus on the right. There, they bind directly to the purple DNA at a specific site called the hormone response element. Finally, you can see the result, the production of the red messenger RNA strand, proving that the hormone has successfully turned on a specific gene. To wrap up our lecture, let us reinforce the most important ideas we have covered today. First, steroid hormone receptors are specialized intracellular proteins that act as direct translators between chemical signals and genetic action, fundamentally altering cell function over hours or days. Second, we learned that their structure is perfectly adapted for this, utilizing distinct domains to bind hormones and grip DNA. Finally, we saw how the receptor occupancy equation allows us to mathematically predict a cell's biological response, revealing that achieving high receptor saturation requires exponentially larger doses of a hormone. You now have a solid foundation for understanding how the endocrine system exerts long-term control over human physiology. Great work today.